Hi, it is Catherine, and I'm here to talk to you about cell communication this week. So Callie and I say it's time to get started. I'm gonna hide myself and let's continue with the lecture. So our question for today is, what is an example of cell communication in the human body? And I want you to take a moment and think about it real quick. I want to hear all about your ideas when we see each other for our class meeting on Thursday. And for the rest of this lecture, the example that we're going to talk about is insulin and glucose uptake. So you probably have someone in your life that's diabetic or pre-diabetic. When I was growing up, my cat was diabetic, so I had a very large cat, and we had to give him insulin shots twice a day to manage his blood sugar levels. It was really interesting, actually. Fun fact, if you give a cat insulin, you're giving them human insulin. We just got it at Costco under his name, which was Paul. He was Paul Creech. So this is a lovely example of cell communication because you're using it all the time. Anytime that you eat a food, like this diagram on the right shows a person eating an apple, you are putting the food into your stomach. You're breaking it up into little bits, you're breaking it up into the monomers. In this case, we're going to talk about sugar because apples have a lot of sugar, so does a lot of the food that we eat. And that sugar is going to enter your bloodstream once it's absorbed through your digestive tract. But the sugar can't actually get into your cells until you release a hormone called insulin. Insulin allows your cells to absorb glucose. So let's look a little bit deeper into this. Insulin is produced in the pancreas. If you haven't taken anatomy or physiology before, if you put your finger like 3 inches above your belly button, that's about where your pancreas would be, but it's deep in there, so it's hidden behind your stomach. In the diagram, your stomach is this organ here, right here. And then this little yellow thing peeking out from behind, that's the pancreas. So deep inside of you. Let's look a little bit closer at it. The textbooks describe the pancreas as fish-shaped. I don't see a fish at all. I don't know about you. To me, it totally looks like chicken tender shaped. So if you've ever had a chicken tender, it kind of looks like that. This is the beginning of your small intestine. And then over here, right next to it, that is your kidney. So all sorts of cool organs next to it. If we were to cut this open and look at it in the microscope, we'd see this, all these specialized cells, which are really neat. These cells are going to be capillary cells involved in excreting stuff that you use for digestion. And then this purple structure, that's called the islet of Langerhans. And inside this purple structure, you've got 2 types of cells. You've got pink alpha cells, which are used to make another hormone that we're not going to talk about. But it works in tandem with these beta cells, the purple ones. They release the hormone insulin. So let's zoom in on this beta cell right here. We've got this beta cell. Here's its nucleus, and it's showing you all of these vesicles. And this beta cell is going to make a protein, which is insulin. Insulin is a protein hormone. It's going to eject that protein hormone out of the cell into the bloodstream. So this is the inside of your blood vessel. When it's in your bloodstream, the insulin will interact with pretty much every cell in your body because blood goes everywhere in your body. Every single cell is within like 3 other cells of a capillary, which are our smallest blood vessels. So once this insulin's in the bloodstream, it literally goes everywhere from your ears to your toes. And some of our cells are going to be able to interact with it, only the cells that have insulin receptors. If a cell does not have an insulin receptor, it just doesn't even know that the insulin is there. It can't see it. So if you have an insulin receptor, it'll bind to the cell, and that will allow your cell to take in glucose. You might be wondering why your cell can't take in glucose without insulin, so let's dig a little bit deeper. Here's a close-up. I've got my phospholipid plasma membrane right here. It's a phospholipid bilayer. Here's one half, here's the other half. All of their hydrophobic tails are facing together. So this is my cell membrane. Embedded in my cell membrane, I have the outside of the cell, inside of the cell. Embedded in it are these insulin receptors. So they're hanging out inside the cell membrane. When insulin is present, it'll bind to the receptor. Once it binds to the receptor, it'll activate the receptor, and secondary messengers will cause some kind of effect. That's what's being shown by these arrows. This is your cascade of communication. The effect in this case is that vesicles containing the GLUT4 transmembrane protein get told to leave their areas where they're hanging out and go fuse with the cell membrane. By fusing with the cell membrane, you increase the number of GLUT4 transporters that are available. The insulin binding tells these receptors to open, and once they open, glucose, a pretty large molecule, can come into the cell. Now, if glucose was tiny and nonpolar, it could just go straight through this phospholipid bilayer. But since glucose is relatively big, it's a carbohydrate, it's got different charges on it, it cannot go straight through. So it has to use one of these transmembrane proteins. And in this case, it's called GLUT4. So overall picture, you need insulin to get the right kind of protein to open up your cell so the glucose can come inside. If you don't have insulin, then you don't have the open protein channels and you cannot get glucose into your cell. If you can't get glucose out of your blood, we say that you're hyperglycemic, hyper being too much, glucose glycemic. If you produce insulin and suck all the glucose out of your blood and then you have low blood sugar, like what happens after several hours of fasting, then you are hypoglycemic. You have too little sugar in your blood. Turns out there's a whole different cascade of hormones to fix hypoglycemia, but insulin is used to fix hyperglycemia. So after you have a snack, you break it down, you have extra glucose in your bloodstream, you produce insulin, and then you suck that glucose inside your cells where you can use it at the mitochondria to make ATP. So the sugar isn't used on its own, but the sugar is used in cellular respiration, which is what our mitochondria do to make the ATP that we'll use. We're going to add in some extra vocab to this picture. This whole thing is signal transduction, and the entire process is a cascade because one thing leads to another. It's a cascading pathway. And we start with our ligand. Our ligand is our signal that's going to be taken to the cell and read by a receptor. So in this case, insulin is the cell signal, it's the ligand. The receptor itself is the insulin receptor. In this case, it's a cell surface receptor, but it doesn't have to be in the plasma membrane. It could be inside of the cell. And if it was inside the cell, it would only be activated by a molecule that's small enough and polar and could just go straight through the plasma membrane like carbon dioxide. Once your receptor is activated by the ligand, you have a secondary messenger, and in most cases it's many secondary messengers. They're the ones that cause the effect inside the cell. Insulin itself, it doesn't ever come inside the cell, it just sits outside. So the message that insulin is bringing to the cell is conveyed through secondary messengers. Those secondary messengers have a bunch of different reactions, and this is a total simplification of the actual process for insulin and glucose uptake. These process here, all these arrows of different proteins interacting with each other, that's your signaling cascade. It causes an outcome, which is the cell response. In this case, the outcome is more GLUT4 transporters that will pull glucose inside your cell. You've probably heard of diabetes. I mentioned a little bit earlier. Diabetes is a disorder when your body is not using or making insulin properly. There's two main types, but in reality, there's like 5 types, but we talk about 2 main types. Type 1 is something that you're born with. Usually you're diagnosed as a kid, and about 5% of all cases of diabetes are type 1 diabetes. And in type 1 diabetes, your body just doesn't make enough insulin. We don't really know why, but we do know that it's a lack of insulin. And as far as we know, there's no way to prevent it. And it's a lifelong thing that you have to deal with. In type 2 diabetes, your body might make insulin, but the cells could be immune to it or resistant to it or just not recognize it. So you make insulin, but your body doesn't use it properly. And this is the one that can develop pretty much any time, especially as you get older or if you're pregnant. There's gestational diabetes. And most of these cases can be prevented with diet and exercise and maintaining correct blood sugar. So if you're pre-diabetic, you're not yet type 2 diabetic, but you're on the path to becoming type 2 diabetic. And it's really, really common. So 1 in 3 people will develop type 2 diabetes. So taking care of your body, making sure that you're eating well and exercising well, and going to the doctor regularly and getting checkups and blood work, all of that stuff is important to make sure that you don't get type 2 diabetes and not treat it, because treatment's really important too. So let's take this information and practice working with it, because you know your exam is this week as well. So here is a practice question for you. This is a beta cell, and it's making insulin. The insulin is going to get used in the bloodstream. What kind of transport is this? So I want you to take a moment and think about it. And if you come up with a really quick 1-word answer, I want you to practice thinking about how to explain it to someone who doesn't know anything about biology. So take your 1-word answer and break it down into little pieces and explain it to someone like, you know, your friend who's totally into art but doesn't get bio. All right, so the 1-word answer for what kind of transport is this could be active transport or exocytosis. But the multi-word answer is, this is a kind of transport that involves lots of organelles and is going to require energy. You're going to make your polypeptide, which is your pre-protein, at the rough endoplasmic reticulum, because it's going to get used outside the cell. Once it is made by a ribosome at the rough endoplasmic reticulum, it'll be transported in a vesicle to the Golgi apparatus. The Golgi apparatus is going to modify your polypeptide, put it back into a vesicle, and then ship it toward your plasma membrane, where it will fuse with your plasma membrane and eject your insulin outside of your cell. We know that moving around vesicles uses energy, so that's why we call it active transport, because it requires ATP. And we know it's exocytosis because we're kicking something out of the cell. Exo being outside, cytosis is cell movement. Cool. Let's do another couple. This practice question, check out these awesome images on the left. This is your cell membrane. This is inside the cell. This is outside the cell, the blue. Here's your protein. Glucose is transported into cells at the same, glucose is transported into cells at the same time as sodium. So we've got our sugar here and our sodium here. What kind of transporter is being shown? This transporter is a transmembrane protein. We know it's a protein because proteins are our transport molecules in the cell membrane. We know it's transmembrane because it goes all the way through the membrane. Trans means beyond, so it goes through the membrane. This is a carrier protein. We know it's not a channel because it's not just a straight tube through it. So this is a carrier, and it's going to be bringing something inside the cell. So it's going to be probably along the edge of your cell. All right, adding some more information. Glucose is transported into cells at the same time as sodium. This causes an increase in cellular sodium. High levels of sodium can lead to hypertension, which is an increase in blood pressure. Explain why this can cause hypertension using your osmosis vocabulary. So, an increase in sodium means that I am taking my cell and adding more solutes to it. If I add enough, my cell may become hypertonic to the solution around it. If my cell is hypertonic, that means that water will try and move into my cell via osmosis. This can cause hypertension because the water in your blood system will be trying to pull itself into your cells, which increases the amount of fluid overall in your cardiovascular system. Hypertension, excess blood pressure. Now that we know that we bring sodium into the cell when we pull in glucose, and we pull in glucose a lot all the time, we need to know how we get sodium out of the cell. And the answer to that is a sodium-potassium pump. Sodium-potassium pumps are extremely important to keeping you alive. So what happens is every time 3 sodium are transported out of the cell, 2 potassium are transported into the cell. Your neurons do this to function. They do it in just like nanoseconds. They're doing it continuously every second that you're alive. And they do it so often that they could use 20% of all of your energy. So you're literally burning a ton of calories by using your brain, which is really impressive. I don't know if you've ever had the experience where you studied really hard and then you got really hungry or really sleepy. That's because you're using a ton of ATP for your sodium-potassium pumps. So if a cell must expend ATP to pump sodium back out of the cell, what is true about the concentration of sodium? If we're using energy to activate a pump, it means that we are pushing molecules against their concentration gradients. So based on this question, we know that there is more sodium outside of the cell than inside of the cell because we have to be using energy to push it out where it's more concentrated. Last thing I have for today's lecture is a study tip. So there was a lot of new information this week. I find that if I get a bunch of new information, sometimes I don't know how to integrate it into the information that I already know. My recommendation is a concept map. Now I like this concept map because it's visually more of a web, which is really how my concept maps end up looking. And it shows that you have to add connectors over the arrows. So if you just add arrows, it's a flow chart, not really a concept map. Both are cool, but trying to connect what you just learned to what you learned 2 weeks ago is a great way to study. And you know that if you can't add it to your concept map, it means you don't really understand it, which means you need to pay more attention to it for the upcoming exam. All right, that's all. Let me know if you have any questions as you study. Please feel free to email me. I love emails, and I will talk to you soon. Have a great day.